the title of my presentation is How the PK/PD of Antimicrobials Impact Their Utility in Different Infections, Success and Failure Stories. These are my disclosures. So by the end of this presentation, you should be able to understand the concept of PK/PD of antibiotics, which I'm sure a lot of the people here in the room already understand that part. Describe how success and failure are determined for antibiotics, and finally discuss prominent success and failures of some antibiotics. And I should acknowledge here the paper by Dr. Ambrose on expert in current opinion and pharmacology about the success and failure, which was a great inspiration to create this presentation. So as everyone here knows, the pharmacokinetic, pharmacodynamic parameters, which were already explained beautifully by Dr. Minogue. So we have the MIC here, we have the Cmax above the MIC, which mainly governs aminoglycosides, the AUC above the MIC, which mainly governs vancomycin, as well as other antibiotic classes. And then finally, the time of the MIC, with which beta-lactams are being controlled. So how is success or failure evaluated? I know Dr. Minogue has already touched on this part, but I'm gonna elaborate a little bit. So we have two different parts to evaluate the success or failure of an antimicrobial. So one part is through the PK/PD perspective. So we have to make sure, as Dr. Minogue explained, that we achieve sufficient concentrations of the drug in the plasma, or mainly at the target site. And not only just the concentration, but also make sure that we achieve the sufficient value amount of the PK/PD index that controls the activity of this antimicrobial, whether it's a Cmax above the MIC, AUC, or time above the MIC. And we should make sure that these values exceed the susceptibility breakpoints of an organism, or even better, certain epidemiological MICs, such as the MIC 90s, using the safe doses, obviously, of the antibiotic, or what would be studied in phase 1 trials that would determine the safety of these doses. And so the higher the probability this target is achieved, increasing the probability of achieving the target attainment, or the PTA. Now, the second part, after the PK/PD, or the preclinical evaluation, we would go into the clinical trials, as Dr. Minogue also explained. So then we would differentiate and find about the effectiveness and the safety of different doses in clinical trials, moving from phase 1, where we evaluate safety, and then phase 2, where we evaluate both effectiveness and safety, and then phase 3 in a larger number of the population. And so usually one of the major primary endpoints in these clinical trials is to find about the clinical success, which is actually more important than the microbiological eradication, which usually falls as a secondary endpoint. And this is quite important because there have been some trials where they showed high rates of microbiological eradication, but less of a clinical success or resolution of clinical signs and symptoms. And this is often have been reported with some Acinetobacter studies. So it's very important to mainly focus on the patient rather than just the labs and what's growing in the culture. So what if there is still no success? What if we're still achieving sufficient concentrations at the target site? Well, it's also important to notice and try to explain if the antibiotic is actually not working at the site of the target site of action. For example, if the antibiotic is being inactivated and despite achieving sufficient concentrations at the site. For example, daptomycin in the lung, tigecycline in bile. And I'm going to discuss these in further details in the upcoming slides. Also, if there is inability to attain sufficient concentrations at the site, despite administration of the maximum safe doses, for example, tigecycline for in bloodstream infections and also in ventilator-associated pneumonia, even though we're giving the maximum safe doses, we may not be I'm talking about the standard FDA-approved doses. They may not be sufficient to actually treat the infection, these sites. Another popular example is eravacycline in the urine. And again, I'm going to discuss each of these antibiotics in details in the upcoming slides. So I'll start with success stories, which are actually very plenty. So I'm not going to delve deep into this. So pretty much any antibiotic that has an FDA-approved indication after achieving the targeted primary endpoint that the FDA specify for this site of infection or sufficient evidence of effectiveness or non-inferiority, as well as the safety for the treatment of different infections. So the examples are numerous. But some unique examples would be, one of the classic examples would be a nitrofurantoin in the urine and beta-lactams in bone, where they all pretty much achieve sufficient concentrations in the bone that are higher than they might see breakpoints of common bone and joint infection pathogens. So speaking of nitrofurantoin as a unique example for uncomplicated for lower urinary tract infections, nitrofurantoin is actually one of the unique antibiotics that is highly concentrated in the urine, achieving up to 60% of unchanged concentration. And the plasma to urine ratio is approximately 1:35. In fact, the plasma Cmax is less to or equal than 1 milligrams per liter. So that really much explains how nitrofurantoin definitely adores the urine, as the slide says. So the nitrofurantoin is available, as you know, in 2 different formulations. So the slower extended release formulation, the microcrystals monohydrates, is actually associated with 30 to 45% higher levels in the urine compared with the normal release formulation or the microcrystals. And the explanation, the hypothesis behind this is that the extended release formulation is assumed to stay longer in the gastrointestinal tract. So this may result in more of the drug is being absorbed compared with the microcrystal formulation. So when it comes, so this is from a PK perspective. So when we put this into a clinical perspective, there was actually while there was no difference observed in the clinical outcomes for the treatment of cystitis, there was actually a significant difference that was observed in pregnancy in pregnant women, where the extended release formulation was associated with a significantly less clinical failure, 7.8% versus 13.4% for the normal release formulation in more than 1,000 pregnant women. Now, moving to discussing oral cephalosporins in the urine. As you can see here, a lot of these oral cephalosporins are highly excreted in the urine. Some of them may be as high as above 90%, while others can be as low as 12%. However, when we put this into calculating it based on the actual dose and the milligram value of the drug that is given, we actually achieve quite high concentrations that are can be higher than the susceptibility breakpoint. So in the CLSI document, the M100 document, you would see that there is actually no specific susceptibility breakpoints for the oral cephalosporins, and this was replaced recently with cefazolin. So as you can see here, actually there is a difference in the breakpoints between the cefazolin that represents cefazolin itself, and this is actually represented based on the high dose of 2 grams Q8 hours. Whereas the cefazolin breakpoint that is used as a surrogate for oral cephalosporins is actually, as you can see here, it's actually higher, and it's actually based on a dose regimen that is much lower than the actual cefazolin MIC breakpoint. And so the explanation or the hypothesis is that the higher breakpoint and the lower dose of cefazolin indicate that an organism with an MIC of any range from 16 or below can be actually treated with these oral cephalosporins. Whereas in other sites, if you were talking about cefazolin itself, you would have a much narrower susceptibility range from 2 and below. So you would need a higher dose of cefazolin. Now moving into some prominent failure stories of some antimicrobials. So the first very popular example would be daptomycin and the lungs, and how the lungs actually beat daptomycin. So the fact that we have reached is that the pulmonary surfactant interacts with daptomycin, making it inactive. Unfortunately, this wasn't discovered until the clinical trial of daptomycin was initiated in comparison with the ceftriaxone in community-acquired pneumonia. And the reason that the company or the sponsor actually moved on with this clinical trial was based on preclinical studies on murine thigh model instead of pneumonia model. So just like Dr. Minogue has explained, it's important to correlate your preclinical studies with the clinical studies. So if you want to study the drug in pneumonia, then the preclinical studies should be in a pneumonia model. But in this case, it was actually in a thigh murine model. So this important part was missed. So if we're going to want to explain this more chemically, what happens is that the surfactant contains negatively charged phosphatidylglycerol that significantly enhances the insertion of daptomycin calcium, which is positively charged. And so this results in sequestration of daptomycin molecules inside the surfactant. And in time-kill studies, daptomycin actually lost its antibacterial activity against Staph aureus, even at 64 times the MIC in the presence of a pulmonary surfactant. And therefore, it's not indicated for pneumonia, even though it's a strong drug for many other indications. However, also daptomycin can be associated with rarely with eosinophilic pneumonia. And the hypothesis behind that is that the daptomycin surfactant interaction may alter the lipid integrity, which may stimulate the inflammatory process, resulting in eosinophilic pneumonia. The second example we have is how the blood kicks out tigecycline. So the volume distribution, as you may all know, as a pharmacokinetic parameter is how much of the drug goes into the tissue. And so the larger the VD is, the less of the drug remains in the blood. So a typical example here is tigecycline, which has a volume distribution of 7 to 9 liters per kg, or around 500 to 700 liters. Whereas other drugs like meropenem or cefepime, they have between 18 to 20 liters. So let's look at this picture over here. So tigecyclines, we're looking at meropenem, for example. So the plasma meropenem concentration is about 50 milligrams per liter. So that's quite high, and it's actually a lot higher than susceptibility breakpoints of about 1 or 2 if you're treating a Pseudomonas or Enterobacterialis. So that's if we're talking about the volume. So this actually calculated as a volume distribution of 20 liters, as you can see here. Whereas with tigecycline, with a volume distribution of 500, the plasma concentration is actually 0.1 milligrams per liter. And so this is definitely a very low concentration. And so where did tigecycline go? It actually could be found virtually in every body fluid, such as bile, epithelial lining fluid, synovial fluid, CSF, as well as other body tissues. So failures with tigecycline were actually observed when the infections are complicated with bacteremia. Now, switching gears to eravacycline, another tetracycline. So the kidneys refuse to release tigecycline. So we saw the example with nitrofurantoin, where 60% of it is excreted unchanged in the urine. The case with the eravacycline is that only 16% of it is excreted unchanged in the urine. And so both the IV dose of 1.5 milligram per kg and the oral doses resulted in comparable plasma and urinary concentrations in the lead-in portion of the IGNITE-2 trial, which was the phase 3 trial for complicated UTI. However, in this trial, the patients were started on all the patients were started on IV eravacycline, and then they were switched to either oral eravacycline or oral levofloxacin. And so at the FDA specified endpoint, which is the post-treatment visit cure, eravacycline was unfortunately inferior to levofloxacin, as you can see here, 60% versus about 67%. And so this is the primary endpoint required by the US FDA as well as the European Medicine Agency. However, the patients who received eravacycline only as IV, they actually had higher response rate than those who were switched to oral. So this is about 54% versus 42%. The fourth example we have is how the bile may impact the activity of tigecycline. So I'm sure a lot of people may actually use tigecycline for intra-abdominal infections, but I'm going to tell you about the caveat here. So this was a study that was done by a German team, where they found that tigecycline's concentration consistently declined over time when it was in the bile, until it reached below the detection limit at 8 hours after incubation, where it was actually remained detectable for up to 24 hours in broth. In terms of time kill studies, tigecycline actually failed to kill Enterococcus faecalis and Escherichia coli in the bile up to very high concentrations, as you can see, up to 1,024 times the MIC with Enterococcus and 256 with E. coli. And so, although tigecycline does actually exhibit very good biliary concentrations that achieve sufficient concentrations in the bile, since actually it has a high volume distribution, these concentrations do exceed the breakpoints of common biliary tract infection pathogens. The findings from these studies by the German team can actually be alarming about using it for simple cholecystitis. Now, what about clinically? So the studies that actually evaluated the use of tigecycline in intra-abdominal infections, they actually used it only for complicated intra-abdominal infections. And so they excluded patients with cholangitis, those with simple or gangrenous cholecystitis without rupture. So only they included patients with complicated cholecystitis, or in other words, perforated cholecystitis. And so in those patients, the bile was actually already released and mixed with other fluids. And so perhaps this inactivation was not really observed. So I tried to look in the literature to find any studies of tigecycline in simple biliary tract infections, but I couldn't find any to explain the in vitro findings. Lastly, we have the example of Ceftobiprole. So it can be used for hospital-acquired pneumonia, but not necessarily for ventilator-associated pneumonia. And so in the phase 3 clinical trial of Ceftobiprole versus ceftazidime, so in HAP, it was actually met the non-inferiority point of achieving clinical success compared with ceftazidime at 77.8% versus 76%. Where the p-value was not significant. And this was in the clinically valuable population. Whereas in the VAP population, it actually failed significantly with a success rate of 37.7% versus about 56% with ceftazidime. And the same results actually were the same for the intention to treat populations and the clinically valuable patients as well. So the authors or the researchers actually, or the sponsor, wanted to further explain the reason behind this failure in the VAP population. And what they found is that the epithelial lining fluid concentration was actually much lower than the plasma. So in one study, was in human, it was up to 15% versus in the murine model, it was 70 to 100%. And so the plasma to, another study, the plasma to ELF ratio was 1 to 0.25 after 8 hours of giving 4 doses of 500 milligrams as 2-hour infusion. In comparison, the ratio of ceftazidime in the plasma versus the ELF was 1 to 0.21, whereas with cefepime it was 1 to 1, but after 2 days of therapy. And so that's why, as Dr. Minogue explained, it's really crucial to measure the drug concentration at the targeted site of therapy. In this case, would be ELF, prior to moving on to the clinical trials. Another hypothesis that was also discussed in this study is that perhaps the critically ill patients in the VAP population, we also had increased drug clearance due to augmented renal clearance. So that might be another explanation as of why the drug did not perform very well in the clinical trials. And so the percentage time above the MIC needed for bactericidal efficacy or activity of Ceftobiprole was pretty much comparable to other antibiotics for Enterobacterialis as well as Staph aureus. And so, as you can see here, we need at least 60% of the drug concentration to be higher than the MIC. And if you're not reaching this amount or this value due to lower concentration in the ELF, or perhaps the augmented clearance of the drug in case of critically ill patients, then you may not be able to achieve that PK/PD target. So in conclusion, the PK/PD properties of antimicrobials can actually shape their future success or failure in the clinical setting. That's why it's very important to design the preclinical PK/PD studies very well. Look for the target site, choose your organisms wisely based on what organisms that are commonly infecting the target site where you want to use the drug. And so the understanding the PK/PD of antimicrobials can provide justification of their outcome clinically in case there was clinical failure. Some of the prominent examples that we discussed was the success of nitrofurantoin for lower urinary tract infections and how it achieves very high concentrations in the urine versus the failures of daptomycin and tigecycline in treating pneumonia and bacteremia, respectively. And with that, I thank you for your attention, and we open the floor for questions.
Thank you very much. This paper is open for questions. I'll start off with a comment then. I think one of the reasons that explained some of the failures in HAP-VAP was not only increased clearance in the ventilator-associated pneumonia patients, but also increased MICs values in those patients.
Oh, that's true. Yes.
And it's the combination of those two things that resulted probably in the failures. And the MICs were massively shifted in VAP patients, which we expect given that they've been in the hospital for such a long time. And if I had, oh, go ahead.
Right. So yeah, that's one of the important points is not only in critical ill patients, not only about the augmented renal clearance, but a lot of times these ICU patients may have already received courses of antibiotics. And so the organisms in their bodies may have, or the infecting pathogens most likely have higher MICs than a non-ICU patient. So that's another explanation. That's right.
And I have one more comment. I'm a big fan of killing bacteria. Right, so 3 logs of killing sounds fantastic, but setting that as a threshold, saying you need 3 logs of killing per VAP for Enterobacteriales, for instance, would remove meropenem or any of the carbapenems or any of the beta-lactams for treating ventilator-associated pneumonia, because none of them get the 3 logs of killing. Mm-hmm. Maybe they get to 2 on a really good day for some isolates, but most of the time they just kind of peter out somewhere around 2 logs. George.
Thank you for a very nice presentation. I just wanted to give you a little bit of backstory on Ceftobiprole. Actually, this is a tale that should not be ignored. At the time that they started their HAP-VAP trial, they had not done an ELF penetration study in normal volunteers. The normal volunteer study, which I analyzed, actually was done when they were 75 to 80% done with their enrollment of the HAP-VAP trial, and you know completely successfully put that up there. But you know the take-home message is, and it goes in with the rest of your presentation. If it's not going to get there, it doesn't kill organisms.
Exactly.
And so it's you know a cautionary tale. You know so. I'll just let it go at that. There's a couple other things, but other people.
Well, thank you very much for the comments. Yeah, I do agree with you. It's very important before testing an antimicrobial in real patients and risking failure and having these patients die out of not receiving active therapy. It's important to take all the precautionary measures, do all the sufficient preclinical studies, ELF measurements, and so forth.
Any other questions, please? Dr. Nicolau, perhaps? No? All right, thank you very much for a very nice presentation.